He originally started working with killer whales in the Pacific Northwest in 1985 and received his PhD in 1994 with a focus on foraging behavior of killer whales while based in Olympia, Washington. And then he is also a member of the Pacific Scientific Review Group, an advisory board of NOAA Fisheries, and is on both the Reserve Advisory Council for Papahanaumokuakea Marine National Monument and the Sanctuary Advisory Council for the Hawaiian Islands Humpback Whale National Marine Sanctuary. That's quite a lot. So without further delay, please help me provide a warm welcome to our guest speaker this evening, Robin Beard.
Thank you everyone for coming. Before I get started, I just want to acknowledge that the work that I'm going to be talking about is a team effort. It involves a lot of different people who are involved in the field projects. They're taking photos, flying drones, tagging, collecting samples, spotting things, recording data. And nothing that I would be talking about tonight didn't involve this big team of people. As well as those that are on the water, we have a bunch of folks back in the office that are matching hundreds or thousands or tens of thousands of photographs, analyzing tag data. And this is a long-term project. We're in our 26th year, so the funding's come from a really wide variety of sources, but most of our funding comes from grants from NOAA Fisheries. And off some islands, like work off Kauai, we get funding from the U.S. Navy. And as well as the team through Cascadia, we have a lot of collaborators, so folks that are doing genetic analyses with various universities or government labs and others. Now, the species that I'm going to be spending most of my time tonight talking about, raise this a little bit. The species that I'm going to spend most of my time talking about are false killer whales. And false killer whales don't look anything like killer whales. They were given the scientific name Pseudorca because they were first discovered as a skeletal specimen. And the skull looks very, very similar to the skull of the killer whale. They're smaller than killer whales and they lack that really striking black and white markings. But they do share a lot of similarities with killer whales. They're both very long-lived, living at least into their 60s, and they're slow to reproduce. The females don't give birth to their first calf until about 10 years of age. Females go through menopause, like killer whales, humans, pilot whales, and stop reproducing in their 40s, but then will live 20 or more years afterwards. They're naturally rare, they're top predators, so they're not really common anywhere. And they also have a very low calving rate. False killer whales are thought to give birth to one calf about every 6 or 7 years. There are some differences from killer whales though. False killer whales are tropical and subtropical, whereas killer whales are found from both polar waters to tropical waters. They're often confused with other species. They don't, like I said, they don't have that striking black and white markings, and so they're confused with pilot whales, and here in Hawaii also with melon-headed whales and pygmy killer whales. And they're primarily oceanic, found in deep ocean waters rather than on the continental shelf as killer whales are. Now, killer whales, you may remember, have this very large dorsal fin in the adult males. They're very strongly sexually dimorphic. False killer whales are also sexually dimorphic, but it's not really in the dorsal fin that's different, and instead it's the shape of the head. So this is an adult male false killer whale, and you'll notice it has a very large nose. Effectively, adult males, their nose overhangs their lower jaw much more than an adult female. Now, the work that I've been doing in Hawaii has primarily been undertaken from relatively small vessels, and these just show a selection of them. I would not recommend working on a boat the size of the one on the upper left there. That was a short project off Kauai, definitely not safe in the typical waters we have in Hawaii. But the boat that we use now, or the type of boat, is primarily this one in the bottom right. It's a 24-foot rigid hull Zodiac. We have a nice bow pulpit on the front that allows observers or photographers to be up high above the water, and it's a safe and stable and fast platform, so it's our ideal research vessel. Now, when we're on the water, we're not following any sort of systematic search pattern. Instead, we try to cover as broad an area as possible while simultaneously staying in relatively calm waters. So we just finished up a project a couple days ago off of Lanai, based off of Lanai, and this shows 5 days of our track lines. You can see 2 days over towards Maui. Those are days that were particularly windy, so we weren't able to work out in the deep water to the west of Lanai. And the rest of our time is being spent as much as we can in the deeper waters to the west and southwest of Lanai. That blue line that's coming in from the west, we actually, 2 of us brought the boat over from Oahu on our first day, and so that's as we're getting closer to Lanai. Now, when we're out on the water, we're working with a wide variety of species of whales and dolphins. I first started working in Hawaii in 1999. I moved to Maui, and when I got here, I didn't want to overlap with other researchers. I wanted to work on species that no one else was working on. So I focused on bottlenose dolphins, which are in the upper left, or upper right, pantropical spotted dolphins in the upper left. When we were able to get out west of Lanai, we would see short-fin pilot whales, and in the Awa Channel, we would see false killer whales. So those were the 4 species that we primarily worked on in that first year. In 2002, started working over off Kona and Oahu, and there we were able to get out into much deeper water, so we encountered a lot more deep water species, and that some of these other ones that are shown here are examples of those. A couple different species of beaked whales, dwarf sperm whales, rough-toothed dolphins, and some open ocean species like Fraser's dolphins. Now, we've also combined this boat-based work with land-based work on a few different occasions, and this was an example from December 2020 when we had a project on Lanai. We had 2 people up at the top of the cliff on the southwest side of the island with a tripod-mounted spotting scope and tripod-mounted binoculars. And they were able to spot whales and dolphins out to about 9 or 10 miles from shore. And so either they could say, no, we have nothing in the area we're working in, so we could go work further away from shore, or they would direct us to something that might be of interest to us. And if you have really good eyes, you can see this is a blow-up of the research boat at the top of the cliff, and you can see a pickup truck at the top of the cliff, and it gives you a perspective of how high that particular cliff is. But we've done the same type of thing off Kauai and off of Kona as well, and it can be quite effective. Now, when we're on the water, we're using a few different research techniques. This fellow in the front of the boat has an air rifle, and on that air rifle is a satellite tag. This is working with a false killer whale off of Kauai. We have 3 different photographers on the boat, and as the false killer whale comes over to the other side of the boat, you'll see him deploy a tag. And somewhere in here, I think I have one of these tags, which I'll pass around. So these tags attach to the dorsal fin with 2 surgical-grade titanium darts. And they anchor on the fin and record where the whale is going for some period of time. Some of the tags, like the one that's being passed around, also has a depth sensor, so it's recording how deep it dives and how long it dives. And one of the reasons why this technique is so important to us is we're really limited in terms of where we can spend our time by the winds, but the whales move wherever the whales want to move. So this is an animation showing movements of, it'll eventually get up to 5 different false killer whales that were all satellite tagged off of Kona. And they're from 2 different social groups, which I'll talk more about in a few minutes, and you can see a few different things from this animation. One is they cover the whole expanse of the main Hawaiian Islands, and they do it relatively quickly. There's a counter that counts by days up at the top there. This is October 16th, October 17th, October 18th. They move from the windward side to the leeward side and back fairly rapidly. And you can also see when there's several individuals that are being shown in this animation at the same time, that sometimes they're together, sometimes they're apart. And that reflects their hunting strategies, it reflects their social organization, and even individuals that we know are part of the same social unit may sometimes separate by hundreds of kilometers around the islands. Now, another technique we use when we have animals close to the boat, question?
It shows that the water around the island, do they stay around the island, they ever go out to the open ocean?
I will get to that in a few minutes. It's a good question, though. So other techniques we use when animals are close to the boat, we may stick a 360 camera over the side of the boat to record underwater behavior, prey sharing, social interactions. And what you can see here, this was taken last week off Lanai, is the fellow crouched over there. Mark Moeller has a crossbow, and we're also collecting small biopsy samples. And these biopsy samples are used for a large diversity of projects. From the biopsy, we get a little bit of skin and a little bit of blubber. The skin is used for genetics. We can say the sex of the animal, who it's related to, whether or not, if we have a lot of samples from one group, we compare to another group and see whether there's differences, more than one population in Hawaii. We can use the skin samples for what's called epigenetic aging to get an idea of the age of the individuals. The skin can also be used to look at stable isotopes, which tell you something about where on the food web they feed. The blubber can be used for histology. There's a grad student over at UH that's using false killer whale blubber to look at body condition. So are the fat cells big or the fat cells small? And that tells you something about how robust the animal may be. We can use it to look at pollutants like pesticides and industrial chemicals. And I'll talk about a little bit of this. We also use them to look at hormones, either reproductive hormones. We can tell if a female is pregnant or if a male is sexually mature based on the estrogen and testosterone in the blubber samples. And this shows one of these biopsy arrows just after it bounced back from this false killer whale and takes a little, as I said, a skin and blubber sample. Now, another main research method we have is citizen science. We have a lot of different people who either work on the water or play on the water that take photos and contribute them. And these include professional photographers, it includes tour operators, it includes commercial fishermen or passengers on trips. And this is particularly important because we come out to Hawaii for field projects that may last a few weeks at a time, and these folks are on the water all the time. So it really fills in a lot of the both temporal gaps and spatial gaps. That said, I mentioned we're in the 26th year of this work. Spend almost 2 months a year on the water here in Hawaii, which is typically a couple different field projects off a couple different islands. So this year we had a project in February off of Kauai, just the last couple weeks off of Lanai, and then we'll have a project in October off of Kona. And since COVID, we've also had what we're calling rapid response efforts. We've been training local people in the research techniques we use, and a lot of our field crew are locally based. And so during COVID, when everything shut down, one of our Kona-based folks basically was going out in the water. He had a boat, he was going out in the water and collecting photos, collecting biopsies, and tagging. And that's been a great addition to what we've been doing. So these little squiggly yellow lines are the vessels tracks over the years. So you can see we've had a lot of effort off of Kona, going out up to about 120 kilometers offshore. We've worked off Maui Nui 11 different years, Kauai 16 different years, and Oahu just 7 different years. And during all of those efforts, we've seen a lot of different species of whales and dolphins. 18 different species of toothed whales and dolphins, and 3 different species of baleen whales. False killer whales have always been our highest priority species for a couple reasons. One, they're just really cool, and I've always been interested in them. Hawaii is an area, probably one of the best areas, if not the best area in the world, to work with false killer whales. And so when I first came here in '99, it was a species that I really wanted to work with. But pilot whales are our most frequently encountered species, followed by bottlenose dolphins. And then when you get way down the list, a lot of species that we see just very occasionally, and that's typically because they're not resident to the islands. They're part of broadly ranging open ocean populations that just pass by the islands. And I have this little note about half of our false killer whale sightings were not because we just randomly came across false killer whales while we're searching, but because we're off Kauai, we're working with the Navy, and they have a hydrophone range off of Kauai, and they can direct us to groups, or we've had tags on animals and we're able to relocate the tagged animals, or folks have just called us and said, we've got false killer whales over here, and so we've gone over as a result. Now this map shows all of our search effort as well as the search effort of Pacific Whale Foundation here at Maui, the Pacific Island Fisheries Science Center, and all of the false killer whale sightings from all of these research sources. And you can see there's relatively few sightings off of Kauai, but lots of sightings off Oahu and Maui Nui and off of Kona, and just a few sightings off the windward sides of the islands that have been primarily the large vessel surveys that Pacific Island Fisheries Science Center has been undertaking. Now, when we find false killer whales, we don't just find one animal, we find groups. Sometimes it's a small group, but other times the groups are really broadly spread over 5 or 10 miles, and it's typically, as you may have seen in that video or the animation, the tag data, sometimes they'll separate and join and separate and join, and so we're often able to encounter multiple subgroups in one day that are all part of this broadly moving, dispersed group of individuals. Now, false killer whales, we're really lucky. We probably know more about what they feed on than any other species of whale or dolphin because they like to catch relatively large fish. And if you're really observant, you may have noticed that right there, there's something sticking up out of the water that is not from a false killer whale, and that's this photo was taken about 10 days ago off of Lanai, and they were carrying a large fish around. And this wedge-tailed shearwater is a good indicator of that. It's actually following the whales around, trying to pick up scraps from this fish. This is another photo of the same whales and the same fish. And for those who may not be able to make sense of this over here on the right is the tail fin of the fish. It's a broad-billed swordfish. This is the anal fin of the swordfish, and these are the pectoral fins. And so what you're looking at right there is the upside-down broad-billed swordfish that these false killer whales were carrying around. And there's one false killer whale on either side of it, so it's just the dorsal fin of it over here. And we're really lucky. It's the first time I had ever actually seen them feeding on swordfish. Because it's such a large fish, it takes a long time for them to feed on. They were carrying it around for over an hour, and a big flock of birds that were scavenging from them. This is footage from one of these 360 cameras that I got 2 weeks ago. And they're carrying part of this swordfish. You can see a couple things. One whale had the fish and then dropped it, and another whale grabs it. You may notice that there were little bits of fish sort of trailing behind as they were breaking it apart. And this also is a good video that shows that they hunt in groups, they feed in groups, they share their prey, and all of those things makes it much easier for us to document what exactly they're feeding on. We see them feeding a lot of mahi-mahi, and it's not surprising for a couple reasons. One, there's a lot of mahi-mahi out there. If you want a good fish to eat locally, mahi-mahi is a good one. They reproduce quickly. They're one of the species that's benefited from the overfishing of ahi. They sort of expanded to start to fill some of that open niche from the lack of ahi. Also, they're really easy to identify if you're not a fish person. Probably the easiest fish that we can say, oh, it's a mahi-mahi. Many of the tunas, there's a lot of different species of tunas. Sometimes it's obvious and sometimes it's not obvious which species. They also feed on reef-associated game fish. This is a lua, giant trevally, and we've been able to document both through our own observations and photographs from citizen scientists, and also working with Christy West in her lab at the University of Hawaii that does examination of stomach contents of stranded animals to document a pretty broad list of what they're feeding on. And I'm going to come back to this in a few minutes, but if you look at this list, one of the things you'll notice is that most of these fish are species of fish that we like to eat. How many people here eat fish? So a lot. Only a couple don't. I think the only one on this list of the fish that people don't regularly eat are probably the filefish. And these photos show a mahi-mahi. The one on the upper middle right is a mongchong, opa in the bottom right. This one down here is a broad-billed swordfish. The other one's a kahala, and then an ono up in the top left. Wide diversity of both reef-associated and pelagic game fish, and I'm going to come back to this later on in the talk. Again, these are all species, or most of them are species that we like to eat, and that's really important when we're trying to understand what factors are influencing the population or what threats. Now, photo identification is a key method that we use. We usually have at least 3 photographers. We take photos of the dorsal fins as well as other parts of the body to identify individuals, and we can answer a lot of different questions. Who travels with who, which tells us something about social organization as well as population structure. Where individuals spend their time. So for example, off of Maui Nui, and I'm going to show you some maps of tag whales, only a couple of the social clusters from this population regularly visit Maui Nui and the other ones don't. We know that from photo ID, but we also know it from tag data. If we get photos of individuals when they're young, we can say how old they are, and then when they start showing up with calves, we can say at what age they might give birth and how often they have calves. And really importantly, with all of the photographs, we can estimate the abundance through what's called marker capture abundance estimation, and I'll talk about that again in a few minutes. Now, this is a social network. When you have those individuals together, I mentioned you can use it both for social organization and for population structure. Every one of these points is an individual, and if there's a line connecting 2 points, it means those individuals are being seen together. So this group that's in pink and around that circle, there are no linkages between that group and any of the other groups. And that means that individuals from that big aggregation of individuals have never been seen associated with anyone else, all of those other, what we call isolated clusters. And all of those individuals in the circle are individuals that were photo identified right around the main Hawaiian Islands. Now, we've satellite tagged 48 different individuals from within that big social cluster, and this shows the density of where they spent their time. So the bright red areas are high density areas, and the white areas are low density areas. And you can see a couple things. One, this is showing a lot of the entire Hawaiian Island chain. That black line, the outer line, is the 200 nautical mile exclusive economic zone. And from the tag data, you can see these whales stay around the main Hawaiian Islands. They've never been, the satellite tagged ones have never been documented moving away from the main Hawaiian Islands. And National Marine Fisheries Service recognized this population as the Hawaiian Islands stock as a result of the combination of photo ID data and satellite tag data. Now, if you look at these other groups that are circled, these are groups that were photographed offshore, either by these offshore surveys that NOAA Fisheries does, or when we've been able to get out to some of the seamounts to the west of Hawaii Island, Jagger and Cook seamounts. And the map shows the movements of those individuals. And instead of being restricted to the area around the main Hawaiian Islands, they're moving broadly into international waters up throughout the northwestern Hawaiian Islands. And these individuals were recognized and designated as the Hawaii pelagic stock, open ocean stock. And you'll notice there are no links, there are no associations between these individuals and that resident population around the main Hawaiian Islands. And I'll zoom out a bit because you can see some of the tracks disappear off to the east. And this shows the full range of those satellite tagged individuals. This one group went about 2,000 kilometers to the east of the main Hawaiian Islands. So it's really truly an open ocean population. Now there's also this other cluster, and there's some linkages and more sightings of those. These are groups that were documented either off of Kauai and Niihau or up in the northwestern Hawaiian Islands. And if you look at the satellite tag data from those individuals, they appear to be a northwestern Hawaiian Island population of false killer whales. And they were designated as the northwestern Hawaiian Island stock in 2012. Again, from a combination of the photo identification data and the satellite tag data. But another really important part of the story and an understanding of the relationship between these groups or among these groups is the genetic data. How many people here have mitochondrial DNA? Okay, everyone should have their hands up. Mitochondrial DNA. This is a lovely greeting card or a Mother's Day card you can get for your mother if you'd like. Happy Mother's Day, thanks for the mitochondrial DNA. Mitochondrial DNA is passed on from your mother to you. She got it from her mother, and your grandmother got it from her mother, and so on and so forth. So what mitochondrial DNA does is it allows us to look at the maternal lineage of a population. And that's really important for a variety of reasons, which I'll get into when it comes to trying to understand the relationship between these three different populations that I just mentioned. So this is a mitochondrial DNA network from false killer whales that have been sampled, not just in Hawaii, but worldwide. When I say sampled, some of these are stranded animals, some of these are animals that were killed in a fishery and the sample was collected, and some were animals that were, we remotely biopsy sampled, as I showed earlier. Each one of these circles represents an individual mitochondrial haplotype. The size of the circle shows how many individuals in this particular sample have that, and the color shows where they were sampled. And one of the things you'll notice is down here in the bottom, what's called haplotype 1 and haplotype 2. If you look at the color coding, those are animals from this main Hawaiian Islands population and from the northwestern Hawaiian Island population. They share the same mitochondrial haplotype, haplotype 1. That's the dark blue and the light blue. And what that means is they came from the same ancestor, the same maternal ancestor. And what probably happened is there was a single colonization event of the Hawaiian archipelago at some point in the past of a group of false killer whales that were all related individuals that found the islands in the middle of the ocean and thought, well, this is nice, we should stay. And over time, those northwestern Hawaiian Island and main Hawaiian Island, even though they share one haplotype, they also have some private haplotypes or unique haplotypes to them. And that indicates that while they came from a common ancestor, they're not interbreeding today. Now, the pelagic animals, the map I showed you and the social networks of all those isolated clusters, they have completely different haplotypes, and there's a really wide diversity of them. And that reflects this much larger open ocean population rather than these groups that came from the same colonization event. Now, I mentioned earlier these clusters, and if you zoom in on that large pink social network and then do a couple different types of analyses, you get these types of social networks. And this is just for the main Hawaiian Island population now. And what it's revealed is that there's 4 different social clusters. Scientists are not very creative. We call them clusters. If someone can come up with a catchier name, that'd probably be good. But that's what we call them because they're really clustered in these social networks. And we've got tag data from different clusters. We have genetic data from different clusters. And the different clusters are doing different things. So the reason why we had this project off of Lanai for the last couple of weeks is because of Cluster 4. Cluster 4, their high density area is just to the west and southwest of Lanai, as you can see here. And it's probably the best place in Hawaii to reliably find false killer whales when the winds are calm. And that's because, as you'll see with some of the other clusters, like Cluster 1, they tend to spend a lot of time on the north shore of Molokai, north shore of Maui, off of Kohala, areas out on the edges of Penguin Bank, areas that are relatively windy. And I'll show one more cluster, and this is Cluster 2. We actually know the least about Cluster 2 because it's really hard to get up to the north tip of the island off Kohala. It's just windy there all the time. We know that that's where they spend most of their time from the satellite tag data. And you can see, for example, cluster 2 rarely, if ever, comes up the outbound channel. They're just not regularly spending their time in the shallow waters of Maui Nui. Now, another reason why I'm very interested in false killer whales, and they're our highest priority species, is because there's a lot of evidence that the populations be declining. The first evidence actually came from Joe Mobley's aerial surveys for humpback whales, and he was recording other species of whales and dolphins while he was looking for humpbacks. And the sighting rate of false killer whales, that graph on the right-hand side, just has shown this precipitous decline from the early 1990s to the 2000s. And on the bottom left here are sighting rates for some of the other species of toothed whales and dolphins. Short-fin pilot whales, spinner dolphins, spotted dolphins, and bottlenose dolphins. And unlike the false killer whales, some are going up, some are going down. It's not a dramatic decline. And that suggests that the decline in false killer whales is real. It's not due to increased wind or differences in sighting conditions among the different years of the surveys. From some of the genetic analyses that I've mentioned, they're actually able to look at evidence for a decline from genetic diversity, and they found that type of evidence. There were sightings off of Maui Nui, published by Pacific Whale Foundation in 2013, that showed a decline in sighting rates here. And there was also declines in sighting rates off of Kona, and an aerial survey in 1989 found very large groups of false killer whales off of Kohala, and much larger than any of the groups that are seen today. And so combined, these various bits of evidence for a decline in the population led the Natural Resources Defense Council, NRDC, in 2009 to petition NOAA Fisheries to list the population under the Endangered Species Act. In August 2010, they released a report that basically documented that the population was a distinct population segment, and there were lots of evidence for this decline. And in 2012, NOAA Fisheries listed this population as endangered. So then the question is, how is the population doing since it was listed endangered? And the short answer to that is not very well. These are abundance estimates from our photo identification work. So each point estimate is basically the best guess, but the length of those error bars tell us something about how uncertain the estimate is. When the population was listed as endangered is circled in red, and you can see that since it was listed as endangered and for a few years before, it's been undergoing a fairly steady decline. Now this is also the results of that same analysis, and it basically shows what's the percentage decline, or how many individuals per year is the population losing. And this graph on the right shows the time since it was listed as endangered. Since that time, it's declined at about 3.3% a year, which means it's losing on average over that period about 5.5 individuals. So there's 55 less whales around today than there were in 2012 from this endangered main Hawaiian Island population. And the estimate of the population today is about 140, so it's a pretty dramatic decline. So why would the population be declining? And really it comes back, I think, to what they're feeding on for a couple different reasons. They're feeding on large pelagic game fish. These fish are among the top predators in the ocean, other than the whales that are feeding on them. And most species of large game fish accumulate high levels of persistent organic pollutants. Things like pesticides, DDT is a classic one, but there's a signal in Hawaii of a pesticide that was used in pineapple cultivation, and also industrial chemicals like PCBs. And every adult male in the population that we've biopsied, sampled, and analyzed for this has levels of these PCBs that could be causing health issues, immunosuppression, and about 1/3 of the adult females have levels that exceed that health threshold. And these toxins are lipophilic. They're stored in the blubber of the whales. And the reason why it's only 1/3 of the females versus all of the males is because the females dump their toxin load when they give birth and nurse a calf. And so the 1/3 of the females that have these really high levels are probably females that haven't reproduced yet, or females that have gone through menopause and haven't reproduced in the last 20 years, and so their levels are high again. In Christy West's work at University of Hawaii, the 3 highest levels in this study are all from stranded animals that were found dead on the beach, and is a good indicator that those really high levels may have contributed to mortality. Pollutants aren't the only problem. There's been a decline, I mentioned it earlier, there's a lot fewer ahi around today. The ahi are a lot smaller. There's some historical data on the average weight of ahi since 1948. In the late 1940s, early 1950s, the average size of the yellowfin tuna caught commercially in Hawaii was about 140 pounds. And I don't know if any of you are fishermen, but catching 140-pound ahi is extremely rare today, and the average size is about half that, and the catch per unit effort has fluctuated, but has generally gone on. There's certainly a possibility that there's deliberate shooting. It's been reported for other species of dolphins in Hawaii. We have melon-headed whales that have bullet holes through the dorsal fin. We have a bottlenose dolphin that's been shot in the head. There are lots of reports of people shooting rock-toothed dolphins. So it's possible that shooting happens, although we don't have any idea of how often it might happen. But the big problem comes back to the fact that false killer whales are feeding on the same species of fish that we like to feed on. So they, if they see an ahi or something else on the end of the line, they'll at least at times go to take that fish. And there's evidence of them being hooked and sometimes dying as a result. This photo on the right is from the longline fishery, and it shows it was taken by an observer of a false killer whale struggling against the line. But when they struggle against a line like that, they often get injured as a result. So these other photos are animals that have these line injuries on the dorsal fin, and those line injuries are caused by struggling against fishing gear. We published a paper in 2015 that showed about 9% of the individuals in the population had these injuries in the dorsal fin. We published a report in 2017 that showed almost a quarter of individuals either have mouth line injuries or dorsal fin injuries. So they're getting hooked in the mouth, and then so they're more likely to get an injury in the mouth than they are in the dorsal fin. And that's why the proportion is a lot higher. This particular individual has a notch out of its mouth, and what you can see there are teeth. And at least one, if not two, of the teeth are broken off. And so that was from getting hooked in the line, struggling against the line, and having it pulled out. Now, where do these fishery interactions occur? We did a study a few years ago where we took all the commercial marine license data from Hawaii and all of our satellite tag data, and we came up with an overlap index to try to say, if you're a commercial fisherman and are catching fish in Hawaii, where are you most likely as a fisherman to have a false killer whale try to take your catch and thus potentially get hooked? And what we found is that this area off of the Kohala and north of Molokai, between Molokai and Oahu, are really the highest overlap areas between individual fishermen and false killer whales. So I think that that's where a lot of the interactions are probably occurring. How many people here are fishermen? A few. There's a lot of different types of fishing here in Hawaii. There's troll fishing, there's palawahi fishing, there's shortline fishing, ikashipi fishing, various other types of handline fishing. And if you look at what they're targeting, a lot of them are targeting species that false killer whales like to feed on. So it's not one fishery, it's probably a lot of different fisheries, and the interactions are probably mostly occurring in those areas that I showed in the last map. Another problem is toxoplasmosis. You've probably heard of this with monk seals. This is a parasite that's carried by cats and shed in the feces, and cats are the definitive host. If there were no cats in Hawaii, there would be no toxoplasmosis. Prior to cats, there was no toxoplasmosis here, and it would probably take many years for the toxoplasmosis to be flushed out of the system. But when there are cats, especially cats that are loose in the environment, they're shedding the oocysts of the toxoplasmosis. It's getting washed into the ocean. It's killing monk seals, It's killed a couple spinner dolphins, and it's been detected by Christy West's lab in at least one false killer whale, although it wasn't responsible for death. But really importantly, only a very small proportion of the false killer whales or other whales or dolphins that die in Hawaii are ever found and wash up on a beach, so that this type of testing could be done. So the two spinner dolphins that have been documented with toxoplasmosis are probably representative of about 40 spinner dolphins dying from toxoplasmosis. Now, going back to the decline and the ESA listing in 2012, I mentioned earlier that the current estimate is about 140 individuals, and right now, NOAA Fisheries is not really doing anything to try to reverse that decline. But there are some, I do sometimes at least have hope, and one of the reasons is when we see newborn calves. This was a calf that we photographed 2 weeks ago off of Lanai. These vertical lines, the pale lines that you can see, are called fetal folds. This indicates that this calf was probably born just within a few weeks. Also, the dorsal fin was a little bit canted, which it straightens up when they're a few weeks old. So this was a very, very young calf. When we see calves, they tend to be really robust, which is good. That means their mothers are getting enough food and transferring enough nutrients to the calves that at least the calves are starting off well. But then the big question is what happens to them in later life. And I'll just end with this video because it's a really nice video taken off the South Shore of Lanai a few years ago, and I would be happy to answer any questions. Hannah.
You need a microphone. Lily has a microphone over there.
Thank you so much, Robin. Always a pleasure to see your presentations. So, I'm curious, for the average person in Hawaii, do you have any recommendations for individuals to how they can help this very serious situation facing this very special, very heartwarming species? How can individuals here, what can they do?
Good question. First thing people should do is vote, and vote for people, vote for politicians that they know or think will do something for protecting the marine environment. So that's fundamental. On a more personal level, I think there's a variety of small things that folks can do. If you're going to eat fish, try to eat fish like mahi-mahi and ono that are really fast reproducing fish, and try to eat fish from sources where you know where it came from and the type of fishing method people use. Those high levels of pollutants are things, the ones in the study that I showed are pollutants that are really well known as environmental contaminants, but there's a lot of other environmental contaminants that are less well documented, things like flame retardants. So if you can afford it, buy a couch that's doesn't have flame retardants in it. Buy organic food rather than non-organic food. All of those things affect not just false killer whales, of course, but wildlife in general. Yeah, I think those are the three main ones that I can think of at the moment.
Thank you, very interesting. I have a question about your photo ID process. Is it like humpback whales where you send a picture to Happy Whale and there's an AI algorithm, or do you have individual graduate students who are IDing?
Yeah, so the matching algorithms for humpback whales work fairly well, in part because of the complexity of the patterns on the underside of the flukes of humpbacks and how rarely that changes. For dorsal fins, there is a matching algorithm out there, but I am not a big proponent of it, in part because it may be fairly good when individuals look exactly the same as they did the first time they were photographed, it may say, or make it much easier to find that match. But what happens with some species like false killer whales is they're acquiring new notches on the dorsal fin on a fairly regular basis. And what that means is that with these matching algorithms, they miss those. And there's a real cost in terms of conservation, because if there's a high mismatch rate, then it makes you think the population is larger than it really is. And so we have humans that do it, and we're really lucky that the people that we have working on our photo ID catalogs are just really, really good at it. And it's like a detective where they may take a whole bunch of photographs all taken at different angles and look for subtle markings on the body that would never be captured by one of these AI matching algorithms. And matching algorithms, so how often individuals acquire notches is reflected by what proportion of the population has notches. So for spinner dolphins, maybe 35% of the population have notches, and that means they're not acquiring notches very often. So these matching algorithms may work well for spinner dolphins, but for false killer whales, it's about 75% of the population that have notches, which means they're acquiring them much more regularly. People like them though because they're flashy. People are attracted to flashy things.
The slide with the cats, could you go over that again? I didn't quite get all that.
Yes.
What is going on there?
Well, there are a lot of cat colonies where people are feeding feral cats in Hawaii. And cats have this parasite, Toxoplasma gondii, that in cats doesn't kill them, but the parasite reproduces inside the cat and they shed basically the eggs. They're called oocytes in the cat feces. And when those oocytes get into some other species, it can cause an infection called toxoplasmosis. And in monk seals, it's actually a fairly serious problem here in Hawaii. Monk seals, when they are exposed to these parasites, and these parasites can live in salt water, or the eggs, the oocytes, can live in salt water for a couple of years. And so whenever there's a heavy rain, and a lot of these cat colonies are near the ocean, whenever there's a heavy rain, basically the oocytes are just washed into the ocean. Toxoplasmosis, I don't know the numbers for monk seals, but it's more than 5 or 10 individuals that have died from toxoplasmosis. They can be exposed to parasites and not die, or the oocytes. But spinner dolphins, it's called disseminated toxoplasmosis, where it basically causes a disruption of most organs in the body and kills the animal. There's been 2 spinner dolphins that have been diagnosed with disseminated toxoplasmosis. False killer whales, you can, if you have a stranded animal and you collect a dead stranded animal and you collect samples from all the organs, you can basically do genetic testing of those different organ samples and determine whether or not the animal has the oocytes or has been exposed, has the actual parasite in it. And there's been at least one false killer whale that's been documented from this population with Toxoplasma gondii infection. It didn't kill it. I don't know how many people in this room have ever traveled in the tropics and have come back with parasites. Sometimes they kill you, but the vast majority of the time they don't. And in the case of this one false killer whale, it didn't kill it, but it rapidly kills monk seals. It is known to kill spinner dolphins, and it probably kills other species as well.
Thank you.
We showed a graph of the decline of the population of the false killer whales. Is there any chance that they're being displaced and going elsewhere besides the Hawaiian Islands? Or is it safe to assume that the population is just declining due to that?
Well, I think it's always possible that animals have left, but I think extremely improbable, and that's based on the satellite tag data we have for this population. None of the animals have gone more than 100 kilometers offshore. The long-term resightings, our catalog for false killer whales started actually in '85. Dan McSweeney over in Kona was taking photos of false killer whales and other species since the mid-1980s and contributed all those photos to our catalog, and it shows these long-term resightings, but also the genetic analysis. The animals that have been sampled hundreds of kilometers offshore have all had these really diverse mitochondrial haplotypes. I didn't mention in the genetic analysis, I showed the mitochondrial DNA, but the nuclear DNA shows that the populations are genetically differentiated, which means that interchange with pelagic populations is so rare that it's insignificant. So I think it's really unlikely. And you know, these are cultural animals. They live here all their lives. I think it would take a lot for them to leave. What happens when populations shrink is that, and this is well documented with many populations, is that animals congregate in what we consider their core areas, so their range shrinks. And I think that's responsible for the decline in sighting rates in the Island Channel. I think it's what's responsible for the decline in sighting rates off of Kona. From our tag data, neither of those areas are core areas or high density areas. And so as the population shrinks, they're just going to spend more and more time between Oahu and Molokai and north of Maui and off of Kohala. And I think that's what's generally happening.
Hi, maybe you said this in different words, but because the pink dots of the population are really only associated with those populations that are in Hawaii, and they didn't really associate with the gray dots outside of Hawaii, is that causing a genetic bottleneck that could have something to do with the decline in populations?
Yeah, it's a really good question, and I mentioned the similarities with killer whales. Killer whales in the Pacific Northwest are this extremely well-studied population. The southern resident killer whales, they were listed as endangered in Canada 5 or 6 years before they were listed as endangered in the US. And in that population, they've not only genetically sampled everyone, but they have very complete records of how many births a female has, how many calves a female has given birth to, and also paternity, how many offspring a male has sired. And there was a paper that I think just came out last year that showed, because they have such good genetic samples, they're able to calculate the inbreeding between two individuals, and between every possible pair of individuals in the population. And the individuals that are more inbred are producing less calves and have lower survivorship. And that population declined from probably 120 to 150 individuals down to about 76 individuals, and has been undergoing this inbreeding depression effectively for a while now. I suspect that it's happening with our false killer whales, but we don't know the answer because there's just a lot less information on who's given birth and that type of thing, which is my opportunity to mention something that I wanted to mention, which is if anyone has photos or know anyone who has photos, and it doesn't matter how long ago they were taken, those photos can help fill in a lot of these gaps. And they're going to improve the abundance estimates, they're going to find out, improve all of the things that I've talked about. And I'm willing to bet, I like to bet $5, I'm willing to bet $1,000 that there are photos out there that we don't have from Hawaii, even from Maui. So if you know anyone who spends time on the water, talk to them and ask them if they've got some old slides or black and white negatives from the 80s tucked away. Also, before I forget, I have these nice false killer whale stickers which people can pick up. And for those who have photos or might know someone who has photos, we have these little cards that tell what type of photos we're interested in and that kind of thing.
Being as difficult it is to get the data that you're speaking of, is there any idea of the calving rate of the clusters or how it has been in the past, if it's declined, or is that just data that we don't have enough information to determine?
Yeah, we've not tried to really assess that simply because we don't have enough information. I will say cluster 4, well, one thing I didn't show tonight is that of the 4 clusters, 3 of the 4 have evidence of a decline. For reasons which remain unclear, cluster 2 has no evidence of a decline, and I don't know why that is. This nice fat, it's not this one, but the one that I showed later, newborn calf in cluster 4. Cluster 4 seems like they have more fat calves than other clusters. In my opinion, we haven't quantified it, and Jens from PWF may be able to quantify that from the photogrammetry work. We've looked at body condition from lateral photos like this, and calves in general throughout the islands from all clusters seem to be in much better, calves and juveniles seem to be in much better body condition. And then when they become subadults and adults, then there's just a lot more variability in body condition.
Do you have, I don't know if there used to be a phone number that we could call to report live sightings of false killer whales. Is there contact information for your organization? If we see them, we can say, like, here we are, and coordinates and that kind of stuff?
Yeah, I think what's happened is there's been some consolidation, and now Division of Aquatic Resources from the state of Hawaii has a number that they're advertising, which embarrassingly I do not have with me here. But they have a number to call, and they are in touch with various research groups. So if they get a call, so the idea was to just have it all in one central location. So the calls go in, and then they'll let folks here on Maui know, for example.
The animation you had of the tagged individuals that was bouncing around the Hawaiian Islands, it was really just neat to watch, and I noticed that all the little dots, they'd speed up at the same time and slow down at the same time. I'm guessing that shows what time of day that they're more active and swim a little less speedy. I was just sort of curious about that.
Well, it's a good question. We have, so some of the tags that we've deployed have the dive data as well, so we can get a little bit better idea of behavior. And I think we have 5 individuals from this population that have dive behavior, and then we have, I think, 3 from the pelagic population and a couple from the northwestern Hawaiian population. And one of the things that's apparent is that they're not all doing the same thing at the same time. Some of them are diving deepest during the day, and some of them are diving deepest at night. And you compare that to some other species like pilot whales, for example, or spotted dolphins that have these very stereotypic day-night patterns. And so we're not seeing that in false killer whales. We haven't, we've looked at some differences in depth used by cluster I think we have done some preliminary analyses to try to see whether there's a diurnal pattern to where they're spending their time, and it wasn't showing anything obvious. Sometimes they're out of deepest water during the day, sometimes they're in shallow water during the day, and it's not really consistent among individuals. And I think that, pilot whales and spotted dolphins are both feeding on prey that are associated with the deep scattering layer, which has this very predictable, it's down deep during the day and comes up to the surface at night, so it's a very predictable prey source. Whereas false killer whales are feeding on this real wide diversity of game fish, and there may be some patterns in some of those species of game fish, but they're feeding on so many species of game fish that I think it means that any one false killer whale or any one group may change their behavior over time. So we saw swordfish, we saw them catch swordfish 2 days in a row off of Lanai, and it was exciting enough seeing it the first day, and then they caught another one the second day. And we had never seen them catch swordfish before, so the fact that they caught them 2 days in a row implied that there must be a lot of swordfish out there, and that's what they were targeting those 2 days, compared to our typical case where we may see them catch 3 or 4 mahi-mahi in a day, or one ahi and then a couple of mahi-mahi. Shannon?
First, I just wanted to share the number for the Have You Seen a False Killer Whale? And this reaches out to the NOAA partners, Pacific Whale Foundation, MMRP, DAR, and Robin's group. So that number, if you're interested, is 888-256-9840. It's 888-256-9840. And then my question was, you couldn't, I've seen other presentations where I think you've said that false killer whales are your favorites. So if you couldn't choose false killer whales, what species is your favorite to observe in the wild and why?
I actually have great difficulty answering that question because I just enjoy seeing whatever we see. And it's always nice to see species that we don't see very often, or species doing new things that we haven't seen. 2 years ago, pilot whales, which were at the top of the list for the species we see a lot of pilot whales. Sometimes my field crew is going, oh, not more pilot whales. We saw pilot whales catching fish in the middle of the day off of Kona, which in 800, that was the first time we'd ever seen it in 800 sightings of pilot whales. So there's always the potential to be surprised. And so yeah, no real particular favorite species.
I used to work on a gill netting boat. It's more regret than ever that they banned that. Are there any kinds of fishing that could be like more monitored or more deleterious than others? Like, I don't think any long line fishing.
Now, long line fishing is excluded around the main Hawaiian Islands, so it occurs, but it's all offshore. There used to be long line fishing right up near shore up until 1991 in Hawaii. But then there was a long line exclusion zone that went in around the main Hawaiian Islands, mainly to reduce conflict between nearshore fishermen and long line fishermen. But other types, hook and line fishing in general is widely considered to be a more selective type of methodology than, say, gill nets, long lines. Sometimes part of the public education efforts, so we've done a couple things, and I only have one of them here with me, but a number of years ago we produced these blackfish identification guides, in part because fishermen in Hawaii said that they would see groups of hundreds of false killer whales, and really what they were seeing were melon-headed whales or pilot whales, and you regularly, just knowing what you're seeing is a good way to try to avoid them. If you know what a false killer whale looks like compared to one of these other species, you might, if you see false killer whales coming by, you could pull in your gear and move somewhere else, versus pilot whales, which are extremely unlikely to ever take anything off your line, although they do sometimes, but it's extremely rare by comparison. Fishing at night means you can't see what's going on around, but I know that people fish at night, in particular Palawahi fishing with squid, to catch ahi. When we fish when we're out in the water, but mainly it's when we see mahi-mahi under some debris and we'll throw a line in and catch a couple mahi. There's some targeted fishing that you can do, but I think a lot of it is knowing what a false killer whale is, knowing that if you see false killer whales, pulling your gear and moving off to the side or moving in the opposite direction where you're less likely to have those interactions. How right now the knowing which fishery is actually resulting in the injuries it's unclear because there's no observers in any of the nearshore fisheries. There's no electronic monitoring in any of the nearshore fisheries. There's a lot of fishermen in nearshore waters, and so putting observers out is just completely impractical. But I think there are some things that could be done. For example, electronic monitoring, which involves video camera systems, could be required for boats that want to fish in those high overlap areas. And it's a relatively small number of boats that fish up there anyways, because it's the windward sides of the islands, and you have to be a pretty hardcore fisherman to fish up there. But at least that would provide some clarity as to how they're getting hooked, where they're getting hooked. Fishermen in Kona will often say, well, we never see false killer whales or we never have a false killer whale interaction. And they're telling the truth because Kona is not a high density area for them. There's a lot of fishermen, very few false killer whales, and so they just don't see them very often there. And instead, it's a much, much smaller number of fishermen that are fishing in these other areas that I think are having interactions.
You always hear about whales getting caught in nets. That doesn't, nets drifting out there like you hear about some.
Not commonly. There's certainly a lot of marine debris out there, and humpback whales and other species can get entangled in that marine debris. In the South Pacific and the Central Pacific, there's actually a fair amount of purse seine fishing, and they will set the purse seine around groups of things like false killer whales. There's been a number of documented cases with groups of 100 false killer whales that are set on deliberately because the fishermen are trying to catch tuna that they think might be associated with them. And this comes back to the early days of the tuna purse seine fishery in the Eastern Tropical Pacific. It's still, in the Eastern Tropical Pacific, they may not be setting around false killer whales, but it's still happening in the Central and Western Pacific, at least occasionally.
All right, well, let's give Robin another big round of applause. So as I mentioned before, we are going to